Today, we are going to discuss protein sorting to mitochondria. Mitochondria are obligatory constituents of almost every eukaryotic cell and fulfill a variety of metabolic functions. The mitochondrial subcompartments consisting of the outer membrane, just look at this diagram, outer membrane, inner membrane, intermembrane space, and the matrix. Each contain a specific set of, in total, approximately 700 different proteins. Most of these proteins are coded for by nuclear genes and are synthesized in the cytosol. They have to be imported in a continuous process into the growing and dividing organelles. Before starting with the protein sorting to mitochondria, I will tell you about protein sorting in general. Protein sorting or protein targeting is the biological mechanism by which proteins are transported to their appropriate destinations in the cell or outside the cell. Proteins can be targeted to the inner space of an organelle, different intracellular membranes, plasma membrane, or to exterior of the cell via secretion. This requires targeting signals. Targeting signals are the pieces of information that enable the cellular transport machinery to correctly position a protein inside or outside the cell. This information is contained in the polypeptide chain or in the folded protein called signal peptides or targeting peptides. There are two types of targeting peptides. Number one is pre-sequences and the second one is the internal targeting peptides. Next is protein translocation. The translation of mRNA into protein via ribosome takes place within the cytosol. If the synthesized proteins belong in a different organelle, they can be transported there in either of the two ways, depending on the protein. One is co-translational translocation and the second is post-translational translocation. Co-translational translocation pathway begins with the N-terminal signal peptide of the protein being recognized by a signal recognition particle, that is SRP, while the protein is still being synthesized on the ribosome. Even though most secretory proteins are co-translationally translocated, some are translated the cytosol and later transported to the endoplasmic reticulum or plasma membrane by a post-translational system. In addition, proteins targeted to other destinations such as mitochondria, chloroplast, peroxisomes also use specialized post-translational pathways. Now we'll start with the protein sorting to mitochondria. First is precursor proteins. The precursors of nuclear encoded mitochondrial proteins are made on cytosolic polyribosomes. Precursors contain targeting sequences of 10 to 70 amino acid residues, which in most cases are localized at the NH2 terminus as cleavable pre-sequences. Mitochondrial targeting signals are rich in positively charged and hydroxylated residues and essentially lack acidic amino acids. Precursor proteins have to maintain a loosely folded conformation after synthesis. Next is pathway for protein import from the cytosol. First step of the import pathway is the binding of precursor to receptor proteins at the surface of the mitochondrial outer membrane. The outer membrane proteins of neurospora with molecular masses of 19 kD called MOM19 and 72 kD called MOM72 have been identified as components that participate in the specific recognition of precursors. Antibodies recognizing the cytosolic domain of these components inhibit the binding and import of various precursor proteins. MOM19 and MOM72 may represent the mitochondrial targeting sequence receptors, each being specific for subsets of mitochondrial proteins. They are distributed over the whole surface of the outer membrane, but appear to be enriched at contact sites between inner and the outer membranes where translocation takes place. Receptor-bound precursors can probably move laterally toward these membrane contacts. The entry of precursors into the translocation apparatus is thought to be facilitated by a common component in the outer membrane, the journal insertion protein, GIP. Just look at this diagram. This is outer membrane, intermembrane space, inner membrane, and here this is journal insertion protein, that is GIP, in the outer membrane. So entry of the precursor into the translocation apparatus is facilitated by this journal insertion protein. Proteins of the matrix, inner membrane and intermembrane space are routed from GIP into contact sites for further translocation. GIP has not yet been identified, but good candidates are outer membrane proteins of 38 kD, that is MOM38 from Neurospora, and 42 kD from yeast. So antibodies to the later component inhibited the import reaction in vitro. The precursor of a fusion proteins that was arrested at translocation contact sites could be cross-linked to the 42 kD protein. MOM38 was found to form a complex with the surface receptors MM19 and MM72 on extraction from the membrane with detergent. So transport of the majority of authentic precursors from the surface receptor to GIP requires ATP or guanosine triphosphate, that is GTP. Protein translocation into mitochondria occurs at sites where outer and inner membranes are in close contact. Next is proteolytic processing. Once translocated across the mitochondrial membrane, the NH2 terminal pre-sequence of precursors are cleaved by highly specific metal-dependent processing enzymes in the matrix. Just look at this diagram. Pre-sequence cleavage, an essential function, is not coupled to membrane translocation but is probably required for proper assembly of the imported proteins. So here two structurally related components cooperate in the proteolytic processing. One is mitochondrial processing peptidase MPP, and the second is processing enhancing protein, that is PEP. Both these proteins are encoded by nuclear genes and have to be imported from the cytosol as precursor, having cleavable pre-sequences themselves. So PEP, it stimulates the catalytic activity of MPP about 50-fold, MPP being almost inactive in the absence of this PEP. MPP also requires divalent metal cations such as magnesium ions or zinc ions for activity, but it is unclear whether the enzyme should be classified as metallopeptidase. Next is protein export to the intermembrane space. Different sorting pathway based on stop transfer mechanism across the inner membrane has been proposed. So proteins like cytochrome B2 are soluble component of intermembrane space of yeast mitochondria, and cytochrome C1 of the BC1 complex, which is largely exposed to the inner membrane space, are synthesized as cytosolic precursors with long complex pre-sequences. These pre-sequences have a bipartite structure and are cleaved in two steps by different processing peptidases. Their NH2 terminal parts exhibit the typical feature of the positively charged mitochondrial targeting sequences. Cleavage by the processing enzyme MPP-PEP in the matrix removes these NH2 terminal segments of the pre-sequences, resulting in the formation of intermediate-sized species. The terminal COH means remaining COOH terminal parts of the pre-sequences contain essentially uninterrupted hydrophobic stretches of 20 residue that are preceded by one of the four basic residues, a motif reminiscent of the leader sequences that target the export of protein across the plasma membrane in the case of bacteria. Also in the case of intermembrane space protein, the COH terminal segment of the pre-sequences direct the export of the intermediate sized species from the matrix back across the inner membrane. This this was shown with the use of fusion proteins between the complete pre-sequences of cytochrome B2 or C1 and passenger proteins such as cytochrome C. So both the authentic proteins and the artificial precursors could be detected as soluble species in the matrix if import reaction was carried out at a low temperature, means at 10 degrees Celsius, or in the presence of metal chelator to inhibit the first processing step. Raising the temperature to 25 degrees Celsius and reactivation of MPP by adding magnesium ion initiated the retranslocation of the processing intermediate across the inner membrane, followed by proteolytic processing to the mature size. Now, for better understanding, I'll explain this process with the help of this diagram. Just look at this diagram. This is mitochondria, outer membrane, inner membrane space, sorry, intermembrane space, inner membrane, and this is matrix. So, pre-cytochrome B2, a protein, for example, has a bipartite pre-sequences whose NH2 terminal part, just look at this diagram. This is pre-cytochrome B2 and its NH2 terminal part, this zigzag line, it is positively charged and contain the information for the targeting into the matrix. The COH terminal part of the pre-sequence corresponds to the bacterial type targeting signal and direct the export to the intermembrane space. So the precursor binds to the receptor first. This is receptor R at the surface of the outer membrane. Then insertion into outer membrane is facilitated by journal insertion protein, as I already told you, GIP. It is an ATP dependent reaction. So translocation proceeds via contact site between outer membrane and inner membrane, right? This part. So, and is mediated by a putative proteinaceous apparatus X, right? So, insertion into or translocation across inner membrane of the targeting sequence requires the electrical potential. Now, matrix targeting sequences are cleaved by these means mitochondrial processing peptidase enzyme in cooperation with the processing enhancing protein, that is PEP. Then newly imported precursor, this one, it is associate in an unfolded conformation with the heat shock protein HSP60, this one, right? So this matrix proteins fold at HSP60, this heat shock protein, it is an ATP dependent reaction. In the case of cytochrome B2, HSP60 and maybe additional factors stabilize the processing intermediate for the export step. So export across inner membrane via a putative machinery, this Y. So a membrane associated signal peptidase SPP cleaves the export signal at the outer surface of the inner membrane, right?